Thank you. When we are measuring the laminar bolt response, it is a typical observation, or when we are measuring it with gradient echo sequence, it's a typical observation that the bolt signal is increasing towards the surface. And the main issue here is that the bolt response is, of course, indirectly related to neuronal response, both spatially and temporally. And for example, in this specific case, we know that the neuronal activity can take quite specific laminar profile, which does not clearly compare very well to what we see in the bolt response. And so then, since the discrepancy between these two responses can be quite large, and if we want to still make the best use of our high-resolution fMRI data and to allow inference about neuronal activity, then what we can do is to basically model the forward transformation using dynamic physiological model. And as I will also show you today, we can then invert this model using experimentally controlled spatial modulation of the laminar bolt response. So I would like to first acknowledge some previous efforts in modeling laminar bolt response. So Irati Marquarquiaga introduced a physiological steady-state model that is motivated by vascular anatomy of visual cortex. In the same year, Jakob Heinzel also proposed a dynamic but phenomenological model that is able to simulate some dynamic features that are also observed in experimental data. And recently, we have introduced a new laminar model which tried to account for better properties of the previous two models, and well, it also complies with mass conservation principles, and it can be seen as extension of our earlier work known as PDCM to mesoscopic resolution. So a little bit more about the model. If we think about the vascular anatomy of the cortex, and here I want to focus mainly on venous side because that's where majority of gradient echo bolt signal comes from, then those small venules are merging into larger ascending veins, and those are then draining partially deoxygenated blood from lower depths into even larger pile veins on the surface. And you can also realize that those ascending veins are increasing their density towards the surface. And it's exactly this anatomical relationship between venulus and ascending veins and the functional role of ascending veins, which we are trying to model with our hemodynamic model. So we represent venulus by parallel arrangement of compartments where each of them basically models dynamic changes in blood volume and deoxyhemoglobin. Next to venulus, we have a stack of compartments representing ascending vein, where basically each compartment receives input or inflow from venulus on the same depth and inflow from the lower depth of ascending vein, and then the blood in the compartment is collected by the compartment above in the ascending vein. And in terms of the bolt signal, then both venulus and ascending vein are contributing. And because we want to invert this model and estimate the neuronal activity, then the entire model is driven by depth-specific neuronal units, which are then followed by feed-forward neurovascular coupling. So here's some demonstration of spatial temporal dependence of laminar bolt response. If we consider that there is equal activity across depth, then we can simulate some basic hemodynamic response features that are commonly observed in experimental data. So for example, the amplitude of the peak is increasing towards the surface, and also there is a small delay towards the surface. And also the size of initial dip is increasing towards the surface, and similarly this applies to post-stimulus undershoot. So in the remaining time of the talk, I will be focusing on basically how can we invert such a laminar bolt model, and especially emphasize on experimental paradigm that makes this model invertible, which means that we can actually quite uniquely separate neuronal and vascular sources of laminar bolt response. So we have simulated spatial temporal dependence of neuronal response that could appear during working memory paradigm. These simulations were closely inspired by work of Emily Finn, who is right now presenting her work in the session on learning and memory. and you can still see her poster today. And during this working memory paradigm, we basically assume that the neuronal responses are generated during a period of encoding and manipulation of stimulus, where we also assume that there is an adaptation of the response with time, and we also simulated that there is a spatial variability in the response across depths, assuming there are two peaks in the lower and upper depths. And then after this stimulus period, there is a response period of non-action or action, where during non-action trials, we assume that the response continue to decay, while during action trials, we assume that there is an increase of neuronal activity in lower cortical depths, which has similar amplitude as the main peak of the response. So now we can use this modeled neuronal responses as an input to our hemodynamic model and generate basically the depth-dependent bolt responses, which we can see here. And you can immediately also notice here is the hemodynamic blurring due to ascending vein, where basically if we focus on the original effect that was present in the deep layers or depths, this one then propagates to the surface, and you might also notice, especially in upper depths, that there we have now a difference in amplitude between the response to, let's say, action and the main peak. So to make those simulations a little bit more realistic, we basically projected those responses on a piece of cortical surface, where we also varied the thickness of the cortex. And then we downsampled these cortical bolt profiles to temporal and spatial resolutions which are common in high-resolution fMRI data. And these are examples of voxel activation maps during non-action and action trials, and including additive noise and averaging over 15 trials. So here is another way how to look at the data, showing you the individual bolt voxel signal as a function of cortical depth. We decided to basically sample 6 depths, and you can notice that now the biggest difference between those non-action and action responses are in upper depths. And another way how to look at the data is to see the full temporal evolution of the depth-specific bolt responses. And in this form, we use the data to invert the model. So when we are talking about the model inversion, we are mainly interested about optimizing the neuronal parameters. So we describe our model using 6 neuronal units, which contains excitatory and inhibitory activity. And in this testing case, we consider that each depth is independent of each other. It means that it's also driven by driving inputs and modulatory inputs that describe our experimental manipulation. And next to those neuronal parameters, we then consider optimization of hemodynamic and anatomical properties of ascending vein, where maybe it's good to point out that those properties can be in our model parameterized independently of number of depths. And this model was then inverted iteratively using variational Bayesian algorithm. So here are some results. We were able to estimate or fit the data quite well. And here we see in more detail those estimated laminar bolt profiles for both non-action and action responses. And by basically assuming or allowing or modeling both spatial and temporal hemodynamics of the laminar bolt response with our model, we were able to estimate the underlying neuronal profiles at 6 cortical depths, which, even though represent coarse representation, a spatial representation of original neuronal responses, they are still quite accurately matched. So if we focus here, and here more in the detail, we can actually recover the original difference in the amplitude of neuronal response during action and non-action responses, which means that we can actually deconvolve the effect of the ascending vein that is present in our both data. So with that, I would like to summarize that there is an apparent spatio-temporal discrepancy between the laminar, neuronal, and bolt responses, and this is mainly due to vascular anatomy and blood draining by ascending veins. And we can actually explain this with a laminar bolt model. I also showed you that we can use the model or that model is invertible, but we need experimental manipulation that targets spatio-temporal variations in the neuronal and bolt responses to do that. And we also see that the laminar model can be very useful for optimization of experimental designs that are intended for high-resolution fMRI data. And if you are interested in this model, you can find out more about it in our preprint, or I will be also happy to see you by our poster. I would like to thank my colleagues, and thank you very much for your attention.